Hi. In this video, I want to tackle a big question. It's a big question in biology, and perhaps a big question just for all of us is, how did life begin? And what I want to do in this video is to create a story, a plausible story or narrative of how life began from non-living materials and how that led to biological evolution and ultimately to the diversity of life that we see on Earth today. And before I begin, I just want to be clear that we don't know exactly how life began on the Earth. The Earth is about 4 billion years old, and life began on the Earth sometime, we think, between 3.8 and 4 billion years ago. And the early life would have been some small microbial thing, something like bacteria or like an archaeal cell. So they'd be single cells and they would be very small. And as a consequence, there is no record, right? These were tiny things that lived around 4 billion years ago, and the Earth has changed significantly since then. So we don't have any physical record of how life began, and probably there isn't one. So it seems likely that we can never know exactly how life began. That being said, we can start to understand how life can happen. So, and that's important as we think about where we come from, how did we end up where we are, and where are we going next. But it's also important as we look to space and we look to other planets to try to find life there, because we want to look at places that have a high probability of having life. So most experiments around the origins of life are trying to explore mechanisms by which life can arise from non-living things. And it may be that there are a number of different ways that life can arise. And even if we don't know exactly how it happened on the Earth, we can really learn a lot about how it can happen just in general, and how it can happen on other planets. And I want to be clear about this as well. We also don't know for sure any way that life can happen. Nobody has been successful at creating life from non-living things in the lab, though this is an area of active research right now, and in my point of view, it is only a matter of time before we achieve success in this effort. So what I want to do in this video, since I can't tell you exactly how life began on the Earth, is to explore some of the likely ways, or one of the most likely ways, that life began on Earth. I'm going to avoid the most complicated topics in this discussion, and I'm going to avoid exploring many different possibilities. Instead, I'm going to present this as one story, and one story which proceeds in a logical, step-by-step manner. Again, I'm not saying that this story describes exactly how life began on the Earth. I just want to give you some context of how life can begin in a way that is simple enough that you can hopefully internalize this, think about it, disagree if you want to, in a rational way, and even with evidence if you like. And these ideas that I'm presenting here are based on our current understanding of how life began. And I've picked what I think are the most likely or the most straightforward explanations in these different steps in the beginning of life. And last thing I'll say before we get into it, although the order of events that I describe in the exact way that it happened may not be 100% correct, you can be pretty sure that many of the steps that I describe would have had to occur at some point during the pathway to all living things. So let's get into it. And I want to start at the end first to give you this big picture point here. I think underlying both biological evolution and the process of chemical evolution, which, as we'll see, led to life, there is a key concept to understand. And I think this puts into context many things about living things and our own lives and our place in the history of life in the universe. Okay, and that key concept that I want to share is that key to biological evolution and chemical evolution are the ideas of persistence and replication. You've probably learned about these concepts in biology, and you know that in biological evolution, you have survival of the fittest. And survival of the fittest is the idea that creatures, organisms reproduce. In other words, they replicate. And what they're really replicating when they reproduce is their DNA, as well as some other parts of the cell which carry internal information, like perhaps the mitochondria is a little bit separate from that. But to simplify this, we'll just say they replicate their DNA. And those which have DNA that is best at providing the structure, the proteins and other structures that allow an organism to be successful, to survive, and later to reproduce, those are the things that are going to persist. They're the things that are going to stay around, that we'll end up having more of. So I hope you can see that these ideas, persistence and replication, are key to understanding evolution by natural selection. But also they're key to understand chemical evolution. So what I mean by persistence and replication is the capacity for either molecules in chemical evolution or organisms in biological evolution to replicate effectively, and the persistence of those molecules or organisms that are most stable or better adapted to the environments in which they exist. And again, this idea underlines both chemical and biological evolution. So let's look at how this can happen. One of the most likely sites for the origin of life on the Earth are alkaline hydrothermal vents deep at the bottom of the ocean. There are other possible places life could have begun, but this is one of today's leading theories. Anyway, these hydrothermal vents have a constant release of chemicals, as you can see here, and I think I have a real picture. Okay, so in this picture here, you can see an image of an actual alkaline hydrothermal vent, and you see that there's a constant release of chemicals, and you have this constant source of energy as well. Okay, so now going back to the diagram, again, you can see there's this constant release of chemicals, and importantly, these chemicals include several different carbon-containing compounds and also a number of hydrogen-containing compounds. There's some other stuff there too. In particular, for our purposes, important as well are these inorganic ions, which as they're being released from the vent, but also the ones that exist lining these pores that exist in the rock wall, can function as catalysts to speed up certain chemical reactions. So these alkaline hydrothermal vents have a constant stream of chemicals, and importantly, they also have a bunch of microscopic pores. So they have little indentations and holes in them, which are somewhere between like 1 and 10 micrometers in size. And they're roughly cell-sized and roughly cell-shaped. So as all of these different compounds are released from the earth through the vents and sprayed out into the water, and some of them spray into these pores in the rocks, you're going to have the formation of different chemical compounds. Carbons will combine with carbons in a variety of ways, and they'll combine with these oxygen molecules and the hydrogen molecules, and there'll be lots and lots of different chemical reactions that take place here, as they do anytime you have a complex mixture of chemicals and a source of energy. So these reactions are going to be happening in a more or less random way. But what you can imagine is that some of these complex carbon-based compounds are going to form at a greater rate than others, and some will be more stable than others. So those that are more stable, that hang around longer, over time will persist more. They will tend to accumulate. We know that not all chemical reactions are going to occur with an equal probability. So some chemical reactions are going to be favored over others. And then some structures that result from these chemical interactions are going to be more stable and consequently hang around longer than the others. In this picture, what's illustrated in the rock pore here is the emergence of the reverse tricarboxylic acid cycle, also known as a reverse Krebs cycle. And the details of this aren't so important to understand for this story. But what they're showing here is that carbon-containing compounds are going to combine in various ways, and that this can happen, and we know from laboratory experiments that can happen in a way that produces a variety of biomolecules, so things like nucleic acids, amino acids, carbohydrates, fatty acids, and so forth. So in this picture, you can see that some of these carbon compounds are getting trapped inside of the pores of these rocks, and you're going to have the formation of various different carbon compounds. As I said before, some are going to form at a greater rate than others, and some are going to be more stable, so they're going to hang around longer. We know, importantly for this story, that nucleotides, like RNA, as well DNA, but for our purposes, RNA is going to be more important, can form through spontaneous reactions. This has been shown in a variety of different experiments, really going back to the original Miller-Urey experiment, where they took different gases that they thought would be present on the Earth in high proportions at the time of the emergence of life, and they provide an energy source, and this, in their case, was an electric current. And if you just combine these chemicals that were present on the Earth at that time, as well as an energy source, you get the formation of all kind of biomolecules quite spontaneously. So we know that these basic ideas can happen. So we know nucleotides like RNA can form through spontaneous reactions. Going back to the idea of persistence, if any of the nucleotides are formed at a higher rate or higher probability than others, and especially if they're being formed at a greater rate than other carbon compounds, then these nucleotides will accumulate. Not only will the nucleotides accumulate, but they will tend to form chains. Because as we know from studying RNA and DNA, nucleotides can relatively easily make bonds with each other. So RNA will tend to accumulate in chains, and DNA will also tend to accumulate in chains. One thing that's really neat about RNA is that RNA is capable of self-replication. It can catalyze the creation of copies of itself or of its complement. So this goes back to the idea now of replication as well as persistence. So the RNA chains that end up being the most stable and the most successful at self-replication are going to become the most numerous. And again, we'll avoid most of the technical details here, but I think you can imagine if you have a strand of RNA, AUCG, for example, that formed spontaneously inside of our rock pore, is going to be attracted to its complement, UAGC, because of the complementary attraction between A and U and C and G, which is just a basic fact that we study in all biology classes going back to middle school. So I've illustrated this here below. Imagine we're looking here inside of our rock pore, and spontaneously we get this RNA chain form. Well, there's a pool of nucleotides that have been forming as well inside of this rock pore. And because of complementary interactions, the U nucleotide will tend to make hydrogen bonds with the A nucleotide, and the C will tend to make hydrogen bonds with the G nucleotide. So this pool of nucleic acids, some of them will be attracted to the chains of RNA that exists inside of the rock pore. Because the nucleotides can readily form bonds with each other, you might then get a chain that is complementary to the template. And when I say complementary, I mean that this is the original template strand, and that this is its complement, and these things will always bind A to U and C to G. Okay, so at this stage in our story, we have made two strands of RNA, and they're stuck together by hydrogen bonds. But we know these things can be separated, perhaps through the application of heat differences or through the turbulence of the water moving about. In any case, there are a number of different ways that separation can occur. But once separation occurs, now you have a free strand of this AUCG RNA, and separately another free strand of the UAGC strand. Now that same process can again repeat, where our new strands can attract the free nucleic acids that are floating around in the interior of the rock pore. And then those nucleic acids will line up and form bonds with each other to make another new complementary strand. So now you can see in this way we have gone from one strand of AUCG to two strands of AUCG and two strands of its complement UAGC. Okay, and again, this is a simplification, but I think you can see it's relatively straightforward that RNA can catalyze its own self-replication. The formation of RNA spontaneously is very important because now in modern times, right, all of life is dependent on information that is stored in a genetic code. And we know that that genetic code is DNA in all living things today, not RNA. But RNA was likely the precursor to DNA as a storage molecule because of its capability to self-replicate. Okay, we'll come back to that, but moving on to some other molecules, we know that other molecular building blocks can also spontaneously form if you combine the ingredients and provide a source of energy. So you can get lipids or fatty acids, as well as amino acids that form spontaneously. Amino acids, of course, are the building blocks of proteins, which are the things, the kind of machines that carry out the processes of life. We also know that spontaneous reactions between the RNA strands, which were formed spontaneously, and the amino acids that also form spontaneously can and will occur. As before, those reactions which occur at a greater probability and which exhibit greater stability over time, meaning they don't fall apart as quickly on their own, they're going to accumulate. When we study the process of translation in modern biology, we look at our amino acids as having tags of tRNA. And the structure of the tRNA is more complicated than I'm drawing right here, but it's going to convey the idea. So this can arise through spontaneous interactions between amino acids and nucleic acids. Again, these reactions are only occurring because of the chemical affinities that these structures have to each other. Lots of other reactions will also be occurring, but the ones that are best, that are most stable, that persist over time, will tend to accumulate. And as we looked at with RNA, those that can also replicate themselves will tend to accumulate, of course, because more and more of them are getting made. So let's see how that can apply to these amino acids. We can see in this picture an amino acid that is spontaneously reacted with a chain of RNA, so it's got a tag that's similar to transfer RNA. Because of the RNA chain or the RNA tag that is hanging off of the amino acid, there will be interactions with the existing strands of RNA that have already formed spontaneously in a way that is dependent on complementary binding, as we saw before. U binds to A and C binds to G. When this starts to happen to multiple amino acids, these amino acids will be brought close in physical proximity to one another. Some of these amino acids will have a structure that is conducive to them binding again spontaneously to one another. This bond can be a strong bond, a strong covalent bond, whereas we know that this complementary interaction between our RNA is due to hydrogen bonding, so it's relatively weak. So you'll be able to pop the amino acids back off of this RNA strand, again through the application of some outside energy, perhaps through heat or perhaps through the turbulence of the moving water. Okay, so then you're going to have a free chain of amino acids. I've made only a tiny chain here of 2 amino acids, but you can imagine you could continue adding these on. So long as the result is a stable molecule, it will tend to accumulate and persist. Okay, and perhaps this chain here is also able to attract free nucleotides from the pool of nucleotides in our rock pore, and so it's going to attract to itself its complement, like I just erased. And again, this complement can pop off, and it may attract again another series of free-floating nucleotides that can then bind with each other to form another strand of RNA. These strands of RNA, of course, could attract more of these tagged amino acids. Okay, so in this picture, what you have are strands of RNA that spontaneously form that attract amino acids and make amino acid chains. Those amino acid chains are able to also attract nucleotides and make the same RNA strands that led to their creation. So they're mutually reinforcing in this scheme. They are both helping each other to replicate more and to exist stably over time. So they are going to tend to accumulate. The most stable ones and the ones that are the best at replicating are going to accumulate over time. It's a similar idea illustrated in this image. They have just elongated more and more their chain of amino acids to become more and more similar to a functional protein. Okay, so perhaps separately from all of this, we also have spontaneous formation of fatty acids or other amphipathic compounds. Amphipathic compounds are very important to the story of cells and to life. What it means to be amphipathic is that you have some part of the compound which is hydrophilic, meaning it's attracted to water, and then you have a part that is hydrophobic. In modern cells, we have phospholipids, and they form into a bilayer, sort of like this. In early cells, it was more likely that we have more simple structures. But what happens when you take an amphipathic compound and put it in water is that it will self-assemble into several different cell-like shapes. And this happens, let's just take the example of this vesicle here, because let's say this whole thing is in water, and it formed in water. So all of the constituent pieces, the constituent molecules, were once free-floating. Okay, so because they're in water, what's going to happen is that these hydrophilic heads, which are attracted to water, are going to face towards the water. They face towards the water that is on the exterior out here, and they also face towards a water that's in the interior in the center of the cell, because they have formed in water. Conversely, the hydrophobic tails, being that they are repelled by water, are going to minimize their interaction with water by facing each other or some other hydrophobic substance. And you can demonstrate this experimentally quite easily if you just take any amphipathic compound, like for example soy lecithin you can buy as a supplement, and you put it in water, it will form these microscopic cell-shaped spheres. So in order to get something like a cell membrane, all you really need is to have the formation of some amphipathic compound and water. All right, in our rock pore, this may have happened inside of our rock pores, which are again roughly cell-sized. And you're going to have these structures minimizing their contact with water by having the the hydrophilic parts facing, let me get a smaller marker. You're going to have the hydrophilic parts facing the water, and then the hydrophobic parts minimizing their contact with water, sort of like this. And I'm drawing this as 2 layers here. It could also be that it made 1 layer instead. Either one could work in this early stage. Okay, and it may be as well that some of the amino acid chains, some of the peptides that we formed earlier, may also help to catalyze the formation of these amphipathic compounds. Okay, so what we've got up to this point is a rock pore that is lined with amphipathic compounds and encloses a metabolic pathway inside of the cell that is producing carbon compounds like RNA as well as amino acids, and these things are interacting with each other. The RNA and the amino acids are interacting with each other. The RNA and amino acids are interacting with each other in a way that promotes mutually their replication and their stability in the pool of possible chemical reactions. So again, they will tend to accumulate. Within our protocell, and I'm calling it a protocell because it is not the same as a modern cell, but already at this stage it is very similar to a cell. We're going to have material accumulating, and we're going to have the protocell grow simply through the addition of the material that is resulting from spontaneous chemical reactions of the compounds being released from the alkaline hydrothermal vent. As our compartment grows, specifically as more and more amphipathic compounds get added to our membrane, parts of it are going to break off. And this is very similar to what happens when you blow a bubble. So let's say this is our bubble blowing wand. As you blow through there, you may get initially this kind of long tube of bubble, but as that tube gets longer, is going to be unable to sustain this length, and parts of it are going to break off, and you're going to end up with spherical bubbles. A similar thing happens with amphipathic compounds when they're in water. So we're adding material to our membrane, and that membrane is extruding outside of the rock pore where there's a lot more turbulence, and eventually this is going to break off. Inside of our membrane, both in the rock pore and outside of the rock pore, in my picture you have accumulation of RNA and amino acids. Of course, sometimes you're not going to have accumulation of these substances in the parts that break off. You may have an empty vesicle like this. Yeah, lots of different things are going to happen. But those things are not going to become the most numerous because they have no mechanism that's going to promote their replication or their stability persistence over time. And so we're going to look at that in a little bit more detail, because at this point you've got something similar to a cell. You have nucleic acids that determine how amino acids interact with each other. So already you have something like the information, like genetic information, that is specifying how amino acids interact with each other, just like DNA specifies the structures. But this free-floating cell here that we have achieved, this free-floating protocell that we have achieved, now has a new challenge. It's in a totally different environment. This free-floating protocell that we have produced has a new challenge. It's in a different environment once it leaves the rock pore, and it's going to be now either attached to the outside of the surface of the vent or floating free in the water surrounding the vent. So the next thing that has to happen is that some of these protocells need to survive their newfound freedom. And this can occur again just due to random variations in chemical reactions. Because of variations in the formation of our RNA strands and the amino acid chains, there are going to be many different combinations in the protocells that get released from the rock pores. Some of those variations, probably most of those variations, are not going to be successful. The protocell will not survive, and the protocell has no mechanism to replicate, so eventually it will just fall apart and make new spontaneous chemical reactions with the stuff around them. But those variations which increase the survivability of our protocells and increase the replication of our protocells, of course, are going to persist, and they're going to accumulate because the other protocells are just falling apart and they don't have any way to replicate if they don't incorporate other molecules inside of them like RNA and amino acids. At some point, the amino acid chains which help the protocell to use the external materials that are in the area surrounding the rocks, these modifications will persist and accumulate. So in this picture, I have drawn a bunch of little protocells in the areas either surrounding the open water of the vents or attached to the rock surface of the vent itself. These are pinching off in a way that we just showed, forming many new protocells. Many of these protocells are going to be unsuccessful and they will simply pop or fall apart. So the environment in the area surrounding the vents is different. You don't have necessarily the same chemicals in the same proportions that you have inside of the rock pore. And you have more of a need to grab stuff from the outside of the membrane in order to continue growing and replicating the protocell. Once you have amino acids, proteins or peptides that are capable of allowing the cell to utilize outside materials in a way that allows them to successfully persist, these things will tend to accumulate. These would be like early transmembrane proteins, and they will help the protocell to grab chemicals that are floating outside that it would need on the inside to form new RNA and new amino acid molecules. Modifications like this, which help the protocell to become more stable, or help the protocell to replicate, will become more numerous. So with some time and through many different random permutations of interactions between the chemical constituents that are being released by the vent, you're going to have the formation of protocells that are able to survive in the rock pores, and they're going to be able to survive in the area immediately surrounding the rock pores. But the ocean is a big place. And in order for this to be an explanation of the origins of life, we have to successfully leave the area surrounding the vents and go into the greater ocean. So leaving the vent cloud is going to be a challenge because there are many different environments that exist outside of that cloud, and the concentration of chemicals is going to generally be lower in the open ocean than it is surrounding the vents. But just like before, we're going to make lots and lots of different variations of these protocells, and those variations that increase the protocell's ability to use outside materials things similar to transmembrane proteins, those which help them to maintain stability, in other words, to persist over time, and those that help them to replicate are going to accumulate. So many protocell variations are going to occur, and those which reliably persist and replicate are going to become most common. This is just a question of probability. Because many different things are happening, those that are unsuccessful will form and die off or break apart quickly, but those that are successful are going to form and stay around longer, and if they're also successful at replicating, you're going to get more and more of them over time. In the beginning, the relationship between nucleic acids and amino acids would probably be a little bit haphazard and may not result in functionally useful peptides or proteins, but again, just because of the sheer number of variations, those protocells that reliably utilize those chemicals to replicate in a consistent manner and create mechanisms inside of the cell and across its membrane that help it to persist and replicate are going to accumulate. And once you have reliable replication that depends in a consistent way on the cell's internal information, in other words, the strands of RNA that it has inside of it, and perhaps some of the other internal features of the cell as well, you then have a process very similar to evolution driven by natural selection. And we can talk more about evolution by natural selection in another video.